En teoría está grabando.
Sí.
No sé, es un poco ahora.
Ah, mira.
Ah, bueno, vale.
Ah, bueno, claro, eso es un...
Él no diga que sí,
Vale.
no grabará. Él va a hablar de mi marcha.
Sí, no. És una així, oi? Tal qual, no se li tradueix a l'anglès, no? No se li tradueix a l'anglès, no?
No creo. Supuesto que, pues sí, la verdad, el idioma tingui al punt. Sí, en inglés. Sí, sí, de vegano inglés. Pero claro, con el grabación, a la ubicación. Bueno, el fum este no se le pot posar amb ell encara.
Ah, vale.
No, però sí. Bueno, sí, posaré la galeria. Per veure més gran.
Vale.
I després, si el pot parlar bé, doncs també. No, però en part tindrem una hora. Perquè tindrem una cosa fantàstica.
Good morning, Paul.
Good morning. How are you, Pedro?
Very good, and you?
I'm okay. It's 8 in the morning, you know. I don't usually give lectures at 8 in the morning.
You look
I gave
what?
a lecture in China a couple of weeks ago, remotely, and it was at 9 o'clock at night. So,
You look
all
great.
right.
So early, so it's okay.
So how is the conference going?
It's going well. Well, just started today, but it's going well. Well, yesterday students made their presentations,
Uh-huh.
and first speakers began today. So there will be a student making you an introduction,
Okay.
and after that we'll start. I think that Katerina are ready. Are we ready?
All right. I'm sorry I couldn't come in person. It looks like a very nice place.
Yeah. There are some students still coming now.
Okay.
Who is going to make the introduction? She has to do from here. Hmm? ¿Cómo? Caterina, Paul can see us. Paul, you can see us.
Yes,
Lead the presentation. Can you see us?
I see your auditorium.
Ah, okay, okay, good. So just make it from. Ah, okay.
Hello? So he could hear you.
Okay. Okay. Hi everyone. It's my honor to introduce Professor Falkowski as our next speaker, and he is coming from Rutgers University. He finished his PhD in University of British Columbia. After his postdoc in University of Rhode Island, he moved to Brookhaven National Laboratory and moved to Rutgers University in 1998. Throughout his research, Professor Falkowski and his team significantly developed our understanding in the environmental biophysics. And his research interests is focusing on the systematically understanding of the electron transformation in the basic chemical reactions and how the energy generation during these chemical reactions, understanding the basic chemical reaction in the organism, molecular metabolic process such as photosynthesis and inspiration. And the protein involved in this process, how the protein structures benefit to help better transform these atoms and how they evolved through the time. And in the end, how these proteins react with this atom basic chemical reaction, how this interact with the environmental change, and how they benefit from help developing the plant and react with the solar system and the extrasolar planets. So now welcome Professor Falkowski.
Thank you very much. Good afternoon, everybody. If you have any questions, it's probably best if you ask them via chat, if you can, during the talk, because I don't want to, if you don't understand some of the things I'm talking about, you'll probably get lost a little bit later. So I'm going to talk about photosynthetic efficiency, energy conversion efficiency in the oceans. It's a topic I've been working on for on order of about 35 years, maybe even more. Let's see here. Next, here we go. So let's just get a little image of the oceans for a start. This is a rather famous photograph. It's from Apollo 11, Apollo 7, I think. And you can see that the oceans cover, and we all learned this in elementary school, somewhere in the neighborhood of about 72–73% of the Earth's surface. And we've had liquid water on the Earth's surface for at least 4.1 billion years. We know that. I'm not going to go into the geological record as to why, but we know this from a mineral called zircons, and we know it from the oxygen isotopes in zircons. We don't actually measure the water per se. So let's just take a look at all that water. It looks like a lot of water, right? And on a surface area it is. But if I took all that water and I collapsed it into a ball, that's the volume of water that would be on Earth, on the surface of Earth. It's relatively small. So to give you an idea as a student, if you think about this, if I represent the Earth's diameter as a meter, 1 meter thick, how thick would the oceans be? Well, it would be equivalent to 2 typewriter pages, not even 1 millimeter. So it's on 200 microns is about the thickness of the oceans compared to the diameter of the Earth. It's a very, very, very thin film. So the oceans are 4,000 meters on depth on average. But the amount of water relative to this volume of rock on the planet is very, very, very small. Now, to give you an idea of how important this water is, though, all the oxygen that you're breathing, every molecule of oxygen on Earth is coming from splitting of water, whether it's on land or in the ocean. Originally, it was obviously only in the ocean. We don't get land plants until about 430 million years ago. We had photosynthesis in the ocean that goes back to approximately 3.4 billion. We're not exactly sure when it started, but it's on that order. Now, in the contemporary ocean, there are modern phytoplankton. So, for example, Many of you are aware of these types of organisms. These are diatoms, for example. This is a very nice picture of a diatom. And you can see in the diatom here, these are plastids, chloroplasts. We only call them chloroplasts in green algae and higher plants. In every other group of algae, this is what we call a red alga. These are called plastids. So you can see there are hundreds of them in this particular cell. And then we have dinoflagellates. This particular dinoflagellate will have one chloroplast or one plastid. And of course, coccolithophores, which are very popular now. So these are the plates of coccolithophores. They're covered with calcium carbonate. And here you can see the individual crystals of calcium carbonate at this very high magnification. And these plates are made inside the cell. They cover the surface of the cell. And when they shed, they go into the sediments. And they are the largest contributor of marine carbonates in the world, coccolithophores, much more than corals. Now, if I take a look at the phylogeny of oxygenic photosynthesis, I'm not going to talk about non-oxygenic photosynthesis in this talk, but here are the cyanobacteria, the blue-green algae. They're the only group of prokaryotic algae or prokaryotic cells that are capable of generating oxygen. And you can see that they're very closely related, or they are the source of plastids. All right. So these plastids were incorporated into various groups of eukaryotes. So we have green algae over here. We have the diatoms over here. We have coccolithophores over here, the haptophytes, and so on. And you have land plants here, for example. So land plants are closely related to green algae, but they're very, very, very far away from diatoms or these other groups of algae. So the algae in general are very diverse. They share a common pathway for photosynthesis, all derived from the cyanobacteria, but they're in different body plans or body forms, if you will. So the body forms are derived from various endosymbiotic associations over geologic time. And here's a very simple way of looking at the symbiotic association. So the original prokaryotic cell contained what was probably a mitochondrion, something like a mitochondrion, which has derived itself from a non-oxygenic photosynthetic bacterium, a purple non-sulfur bacterium. And it took on then this plastid, and it became the first group, this is the proto group of eukaryotes that are capable of oxygenic photosynthesis. And there are three main clades of them, the green algae, of which one single cell went on to become the generation of all higher land plants. So that's an amazing thing if you think about it. Then we have, so that is a primary symbiont. One of these went on to become a group, a very small group of algae called the glaucophytes. I'm not going to talk about them. And then the third one went on to become a red alga. So there are various red algae. Most of them that you're familiar with, for example, are seaweeds like nori in sushi. That's a red alga. But many of them are single-celled. And this single-celled alga is also, red alga is also a primary symbiont. But then as we move along, that alga was incorporated into other host cells. So this was a secondary symbiotic association, and it gave rise to diatoms, the coccolithophores, and some of the dinoflagellates. The dinoflagellates have several different derivations. But these are now secondary symbionts. So when you're talking about a coccolithophore or a diatom, there are in those two cases, for example, 4 membranes around the plastid. 2 from the original cell. So the plastid has its own membrane from the original cyanobacterium. And as it was incorporated into its host cell, a secondary envelope membrane was around the plastid that it was derived from the membrane of the host cell. And then 2 more times that happened, and you got 4 membranes around the envelope of the plastids in dinoflagellates, in most dinoflagellates, in coccolithophores, and in all the diatoms. Now let's take a look at what goes on inside those plastids. So in the plastids, there is a chlorophyll molecule, and this is most, or virtually all, not all, but virtually all photosynthesis is derived from the molecule chlorophyll A. So there's chlorophyll A, chlorophyll B, chlorophyll C. Chlorophyll B and chlorophyll C transfer their excitation energy to chlorophyll A. So ultimately, all photosynthesis is derived from the absorption of light by chlorophyll A, either directly or through other pigments which help it absorb. And this is the absorption spectrum of chlorophyll A, and I just show it to you here in colors. So chlorophyll A can absorb blue light pretty well. And all the excitation energy that is absorbed by the blue is transferred to this band here. This is the what's called QY band, which is a red band. And as you'll see later, the red band can fluoresce. The blue band does not fluoresce. The excitation energy moves from here to there within the molecule on order of about 10 to the minus 15 seconds, a femtosecond. Very, very, very fast. There's no photosynthesis that occurs because of the blue band per se. It's all happening down here. Now, the ocean is blue. So why is the ocean blue? And I'm just going to tell you very quickly that the ocean is blue because of what's called fluctuation density scattering. So imagine 2 incredibly small boxes of water that are right next to each other. In one moment of time, a molecule of water from one box can jump into the molecule, into the box next to it. So one box has more water molecules for an instant than the box that was next to it. And then it can jump back. It can jump back and forth and forth and back and back and forth, okay? So when that happens, the optics of the water molecules of the little boxes are changing. The refractive index with the more water molecules is a little bit higher for an instant, and then it gets a little bit lower as it loses its water molecule. So this is what we call a fluctuation density difference. It's not because of Rayleigh scattering, because of the sky. You could have filled the ocean with vodka and it would still have this property. So the ocean would always be blue if it's a clear liquid. All right, so it is blue. And if you go back for a second, you can see that the chlorophyll molecules absorb blue light, which is why they evolved, or why that molecule evolved to be most efficient as absorbing the blue light. So if we then look down, we can determine to first order, it's not linear, but it's a reasonably good correlation. The concentration of chlorophyll is related to the amount of green light relative to blue light that is coming back to an observer from space. So if you're looking down at the ocean, actually, if you simply had nothing in the ocean except water, it would be blue. But as you add chlorophyll molecules, or phytoplankton, let's put it this way, if you add cells, it absorbs the blue light. And when it absorbs the blue light, the water simply gets darker. So you can use this type of relationship to quantitatively determine from space the chlorophyll concentration everywhere. And so we have satellites that, in this particular case, we have two big satellites that are in space now. We make much smaller ones now, but these were launched several years ago, over a decade ago. They're as big as a bus. They have solar panels to get them power, and they sweep. And so they can cover the entire planet about once every 3 days. And there are different sensors on them, so they sweep both the ocean and the land. They use different sensors for land and in the ocean, because we don't use that algorithm of blue-green to measure chlorophyll concentration on land. We use a totally different algorithm. Now, we can take this green band and blue band and derive global maps of chlorophyll concentration. So we have climatologies of these actually going from 2002 up until present time. These satellites are still operational. Now, years ago, my postdoc at the time was Michael Berenfeld, one of my postdocs. And we did something relatively simple. We took an area of the world where we had thousands of radiocarbon measurements of productivity of the world. And we put those radiocarbon measurements through depth into a simple, this is actual depth model. And then we put it into what's called an optical depth. So in an optical depth model, the 1% light depth is 4.6. That's the E-folding of light. And you see we have a hair straightener, right? So the physical depth means much less than the optical depth. So once you have a hair straightener, then we have to collapse about 2.5 orders of magnitude of this productivity at the surface. And most of that is related to temperature, not nutrients per se, but temperature. So it's normalized production. And everything we do is normalized to chlorophyll, because that's the variable that you can see, or parameter you can see from space. All right, we can make a model, and then we can take this model and we can drive it. In this case, we drive it in the atmosphere and in the land and the ocean, and we can make productivity maps. So I did this with my another colleague, Chris Field, who was then at Stanford University, and his postdoc, Jim Randerson. So Jim Randerson and Mike Berenfeld and Chris Field and I made a little calculation of the productivity on the land, productivity in the ocean on a global scale annually. And this is the result, which is published in 1998, I guess. So it's an old idea, but it's still very valid. So this is the story. You get this productivity to biomass ratios. And from that, you can get a turnover time. So let's take a look. So here's the biomass in petagrams. So the total carbon in petagrams of the marine ecosystems is on the order of about 1. On land, it's on the order of about 600 because most of that is wood. It's not photosynthetic. Now, our calculation is that we get about 48%, 49%, 49 petagrams of roughly 46% of the productivity is occurring in the ocean, and it's turning over about once every 5 days. So that means, on average, a phytoplankton cell lives about 5 days before it has to replicate itself. Remember, they don't make seeds, right? So there's no generation here based on like an embryophyte on land, where you make seeds and you cast off thousands of seeds and a few of them grow to be another tree or another grass or whatever. All right, so here you have the 600, and the productivity is about 54% of the global mean. So it's about 56 gigatons, petagrams. And its turnover time is on the order of a decade. And it's only that low because we have around 30% of the productivity on land is grasses, which turn over annually. If it was trees, this number would be on order of about 20 years. Now, let's talk a little bit about getting to the efficiency issue, which is what I want to talk about for the rest of the talk. So we have an equation which was derived by the late Warren Butler, who was a biophysicist, worked on photosynthesis. And actually, he spent the last 20 years of his career at the Scripps Institute of Oceanography because he really enjoyed San Diego. So we have this capital psi, or the maximum efficiency, which this number is 1. So all of these are rate constants. So these are the 3 mechanisms by which an absorbed photon can be used for photosynthesis, can be emitted back into space through fluorescence, or just dissipating as heat, thermal dissipation. So the 3 terms. The rate constant for photosynthesis is photochemistry, rate constant for fluorescence, and the thermal dissipation. These 3 terms have to add up to 1. So if you know any 2 of them, you can derive the 3rd. You with me so far? So we can measure 2 of these very precisely and instantaneously, in effect. So I can measure the photochemistry of the efficiency and the fluorescence efficiency using 2 different types of techniques. So let's talk about the one that you're probably more familiar with, or should be, if you study photosynthesis on the sea at all. And that is the change in fluorescence FV over FM. This is the variable fluorescence over the maximum yield of fluorescence. That is Kp. So for example, let's just take a look at how we do this in my lab. We have little, what we call flashlet numbers, or this is time. We can do it without flashlet numbers now. So this is the initial photosynthetic fluorescence. This is fluorescence FO. That's the teeniest little bit of fluorescence when you start the measurement. And if you then add a saturating single turnover flash, the fluorescence rises to a maximum value, and that's FM. And the reason this is happening is because we're reducing the primary acceptor to QA minus. This is a quinone. You don't have to know those details. And then you can go through various other things and you can get various other parameters in space. I'm not going to bother you with this. But we have these are our instruments now, in their various configurations. So this is an instrument we use at sea. Here it is in my lab, but this instrument is about as big as a desktop, a laptop computer. It's a little bit heavier, but you just plug this into an incoming stream from a ship, and you can get FV over FM every second of every day for thousands of kilometers. We can also do this with corals, so we have a diving version of this. And you can put this on a remote glider and just dump this into the ocean unmanned. So this can go through a hurricane, for example. And for example, here we have an old use of this type of instrument. So we're going from Portsmouth to the Falkland Islands. This is a ship that is supplying the British Antarctic group with supplies. The Cook, James Cook ship is the name of the ship. So you can see it just, this is the route, a few thousand kilometers. And here's FV over FM. So you can see the FV over FM can go up to as high as about 0.65, and it can be as low as 0.2, or even a little bit lower. And there's a day-night variation in this, which I'm not going to bother you with. This is called non-photochemical quenching. But you can see this is the photosynthetic efficiency across this entire route. And here's light, by the by. So with these parameters, we can calculate the productivity. Now, I want to show you an example of using this in a experimental way, not just survey. So this is an old picture. It's doesn't matter how old it is. It's valid today as it was 20 years ago or 40 years ago when it was first made by Joe Reed. This is a map that was shown originally by a chemical oceanographer at Scripps called Joseph Reed. So here's the surface nutrients of phosphate in this particular case. It could be nitrate, doesn't matter. And in the summer, even in the summer, there's excess phosphate in the subarctic Pacific, in the eastern equatorial Pacific, surround the Galapagos, and then, of course, in the Southern Ocean. The Southern Ocean has excess nutrients throughout the entire year, even in the summer when, austral summer, when the sun is shining quite brightly. Now, the late John Martin finally got enough money together from the National Science Foundation to make ultra clean techniques at sea, so he could measure iron and manganese and other metals without contamination from the ship. And this is the iron concentration in the North Pacific. So this is about 1 picomolar, 100 nanomolar, I mean. Extremely, extremely low concentrations of iron. And John thought about this for a while, and he asked, where does the iron come from? So for most of the world's oceans, there's some that's coming from rivers, obviously, but for most of the world's oceans, iron is coming from land. And for example, here's the Straits of Gibraltar. You're somewhere over here now. Okay, this is Portugal, Spain, and various countries in North Africa. So this is not a false color picture. This is the actual color image of the dust blowing off of the Sahel. That's not the Sahara, that's the Sahel, below the Sahara. This is ultrafine dust that goes across the Atlantic Ocean, for example. So the Atlantic is a small ocean, and it is saturated with iron relative to the Pacific, which is why, if I go back, you see the Atlantic Ocean doesn't have excess phosphate in the summer. Now, because we have an asymmetry in the distribution of continents, the northern hemisphere is much more enriched in aerosol iron than the southern hemisphere. So the south is very low in iron, and especially when you get down to the Antarctic, there's virtually no iron flux. All right. So I'm going to show you how we can measure variable fluorescence, FV over FM, when we add iron to the ocean. So we took 2 ships. This is an old movie, but we've done this 13 times in the ocean. We meaning the collective oceanographic community. And here we're leaving Christchurch, that's in New Zealand, and heading down to the Southern Ocean. And here is just the design of the experiment. Now we're going to add iron as iron sulfate to a big mixing container. So these are our test tubes of iron. Okay. And the mix master never smiles. That's the guy in this shirt here. All right. So he's turning it on. Okay, there's the iron going into this big mixing vessel. He's adding a little bit of a tracer so we can measure where it is. And here's the very expensive delivery system. It's a hose with a lead weight. We're going to kick it off the fantail of the ship. Goes into the ocean. Turn it on. And now we're adding iron. And this is like mowing a lawn. You go up and down and up and down and up and down and up and down, about 8 kilometers on a side. So you're making a square. And I'm just going to show you how we sample. So this is, for those of you who have never seen these kinds of techniques, this is what's called a rosette. This is electronically controlled inside the ship. There are a bunch of instruments on the bottom here. So this is telling you pressures, salinity, depth. Well, pressure is depth, temperature, and chlorophyll fluorescence, not variable fluorescence, just the amount of chlorophyll that's in the water that fluoresces. And here we're telling the winch master to lower it. And these are the clean tools. These are the bottles that are acid washed, kept in a separate area. All of this is covered with plastic, so this is for metals. Okay. And if you're an oceanographer, this is what you do. You draw samples of water. That's my former student, Sasha Totsi. And this is an old variable fluorescence instrument that we had on the ship. And of course, you have to keep it cold. Now we don't do that anymore. We have much more sophisticated instruments, but you can measure variable fluorescence. All right, I'm going to go on. So let's just take a look at how we do this. So here's where we started to add the iron. So you can see we went up and down and up and down and up and down, and then we ended the iron addition here. So this block of water is moving. It's in the ocean. And we can trace where it is from sulfur hexafluoride, which is only a man-made tracer. So that's how we know we added the iron, and we can measure the sulfur hexafluoride virtually instantaneously. All right, now we're outside of the iron addition area, and we're going to go into it. So you can see, this is quite clever, actually. We know how far back in time we were. Okay, so this is a kinetic plot. We can convert that into a kinetic plot. And here's FV over FM as time since iron enrichment. So this is outside of the iron enrichment, and the FV over FM was very low. It's 0.25. And within 24 hours in this particular case, after adding the iron, it rose to about 0.5. And then after maybe a couple of days, 3 days, it goes up to about 0.55, pretty high. So the iron added increased the efficiency by over a factor of 2 within 24 hours. And then it rose to even a factor of about 2.5. So this is clearly, iron is limiting the ability of a cell, in this particular case down here. It's limiting the ability of the cell to use sunshine to generate electrons. And this is the cover of Nature from that experiment. And this is FV over FM. This is not chlorophyll. This is FV over FM in the iron enriched patch. This was for the equatorial Pacific. All right, now let's talk about solar induced fluorescence. And we're going to go to the second term there, which is KF. So the sun shines on the ocean, and it also allows the chlorophyll to fluoresce from space. You can see it. And there's a detector on the two so-called MODIS satellites. And they are going in opposite directions around the Earth. So one has an equator crossing time at around 10 in the morning local time, and the other has an equator crossing time around 2 in the afternoon equator crossing time. So let's see how this works. Now, because of fluctuation density scattering of the ocean, the change in the spectrum of light of pure water goes down as a function of wavelength to the minus 4.3 power. That's an empirical phenomenon. It's not calculated, it's empirical. The sky has a 1 over 4.0000 value. That's the Rayleigh scattering function. Now, okay, so let's just take that scattering function. So here it is in the idealized term, okay? So this is bluer light and this is redder light down here. Now, if there's chlorophyll fluorescence, the chlorophyll fluoresces at about 685 nanometers. It pushes light into space over and above the normal decay function. So this is the fluorescence line height. So this is proportional simply to the amount of chlorophyll molecules in the upper ocean that are being stimulated by the sun. And you can only observe this when the sky is clear. Now, if I divide this by the chlorophyll concentration, which is independently determined from the green-blue ratio, if you recall, that's the fluorescence per unit chlorophyll, and that's called the fluorescence yield. And that's K0. KF, I mean. So how do you determine, we can determine that from space, how do you determine that in the ocean? And just to see what it looks like in space, these are the fluorescence yields, and when the fluorescence yields are high, it means the cells are not very photosynthetically active. And when it's low, they're much better off. They're happier. Let's put it that way. So we have maps of solar-induced fluorescence every day of the year. They're compiled by Mike Behrenfeld himself now, who is at the Oregon State University in Corvallis. And here are the maps on averaged for months for 11 years. All right. So how do you determine what these measurements mean? So I turned to one of my colleagues who has been working with me for 30 years, Max Gorbanoff. And I said, Max, we have to make an instrument that can measure fluorescence lifetimes. Now let's talk about fluorescence lifetimes. It's well known for many, many years that fluorescence lifetimes are directly related to the quantum yield of fluorescence. And it's related to a very funny phenomenon. So you remember that that red band of chlorophyll way back earlier? If all of those, what does that chlorophyll absorption band mean? And why would it fluoresce? That red absorption band means that light has populated higher excited states for the electrons. And when some of those electrons fall back to their ground state, they fluoresce. They give off light. So let's do the following calculation. It can only be calculated. You cannot measure this. Imagine now that you know the population of excited electrons, and we're getting into a little bit of heavy physics here, but if they all fell to the ground state and there was no other pathway for them to dissipate their energy except biofluorescence, what would the lifetime of fluorescence be? That would be the half-time of the decay of the fluorescence. And that is what's called the natural lifetime, tau zero. Now, tau zero was calculated in the 1950s by the late Steve Brody when he was a graduate student. And it was a part of his PhD thesis, actually. And it turns out to be 15 nanoseconds. 15 nanoseconds, a very long lifetime. And tau is a measured lifetime. So normally tau in a very, very happy cell is very low. It's on the order of about 0.3 nanoseconds, 300 picoseconds. And it can go as high as, in very unhappy cells, about 2.5 nanoseconds. So we can measure these. This is just showing you when cells are very, a maximum fluorescence yield. The lifetime is long, and at lowest fluorescence yield when you have the ability of the cell to push the electron to another part where you can use it for energy. The lifetime is very short. So we can build a picosecond lifetime instrument, and we can put this on with our FV over FM instrument. So we can measure lifetimes and FV over FM simultaneously, and then we can take this around the world. So these are fluorescence lifetimes. And to make a long story short, the average lifetime is about on order of 1 nanosecond. So if I have 1 nanosecond is tau and tau zero is 15, that means on the order of about 7%, 1/15, 7% of the absorbed excitation energy is going to fluorescence. So that's the K F on average. Okay. So the average quantum efficiency, that's FV over FM, for the world ocean is about 0.35, which is only about half of what its maximum value would be in a very happy culture. And then I told you the maximum, the average quantum yield for chlorophyll fluorescence is 0.07. So 0.07 plus 0.35 is about 0.42, 0.4. So if I just take that and I put that back into my master equation, and I'm asking how much of this goes to thermal energy, about 60% of the photosynthetically active solar radiation that is absorbed by phytoplankton is converted to heat. This is a pretty low efficiency, but that is the efficiency of the real world. The real world is not acting like a phytoplankton culture that you're growing very nicely in your laboratory at all. So my conclusions are for this talk that most of the time, phytoplankton in the real world are extremely inefficient in converting solar energy to photochemical energy. The limitation is primarily imposed by nutrients, such as, for example, iron. And I didn't show you this experimentally, but we also, in some parts of the world ocean, like the North Atlantic, if you add nitrogen, not phosphorus, but nitrogen, you can increase the photosynthetic efficiency. Now, to get this back to the idea of Margalef. So you're all familiar with the Margalef mandala, I would hope. The way the real world ocean works is phytoplankton, what I call body surf. They sit out there in a low nutrient environment most of the time with low photosynthetic energy conversion efficiency. And at some point, like with a storm or with an eddy, there's a pulse of nutrients that comes to them for a day or so. And so they can temporarily raise their photosynthetic efficiency to close to the maximum for a short period of time, for a few days. And then they go back and sit back and wait for the next wave. That's the body surfing. So this model allows a huge number of different organisms to take advantage of this pulsing of nutrients that is occurring on timescales of days to, in some cases, weeks. And with that, you can get higher diversity of phytoplankton. You can get some organisms like diatoms that do very, very well when you have this type of situation. And some organisms that don't do so well, like Prochlorococcus or cyanobacteria in general, which have no ability to absorb those nutrients. So they just sit out there in the background and they're always waiting. There's no wave for them. There are no cyanobacterial blooms, really. The blooms are occurring with diatoms for the first major part, and then to some extent for coccolithophores. And Pedro did those experiments in my lab, actually, where we took and added nutrients and different rates. And you could compete a diatom and a coccolithophore where if you added nutrients very quickly, like with multiple storms, the diatoms won. And if you turned down the nutrient supply and just added nutrients like once every few days or even less, the coccolithophores win. So I'm going to stop. And I'll take any questions anybody might have. Any questions? I can't hear you.
Any questions then in the audience?
You're all quiet. All right, I gave Pedro or Katerina, I gave you a review, which was published earlier this year in Annual Reviews of Marine Science of this entire lecture. And so if you want to read in more detail or look at references, for example, the Butler equation and how we got the term fluorescence. Fluorescence is actually named after a mineral by G.G. Stokes. Stokes equations, I guess you're familiar with. All right, so
Actually, we have a question.
okay.
Hello, nice talk. My question is about how is expected to change in the future this efficiency in terms of photosynthetic. I don't know if you.
Yeah, so you want the past, present, and future. It's not going to change very much. The oceans have been much warmer in general than they are now. So when the ocean gets a little bit warmer, the thermocline will be a little deeper, and it'll require a little more energy to bring nutrients up to the surface. But you'll also have higher wind speeds because the equator to pole heat gradient will be much greater. So I would say that you're still going to have this body surfing phenomenon. It may the distribution may be a little bit different, but I don't think it's going to be that different. Now, if you want to talk about wind, and I think that's an interesting phenomenon, and I'm a little bit sorry I was discouraged from doing this calculation. Well, I did the calculation, but Suki Manabe, who, as many of you know, shared the Nobel Prize this year, actually, just this past year, in physics for climate change. Suki came to visit me when I was still at Brookhaven National Lab, and I had been working on a past issue of how can you tell what the productivity or the chlorophyll concentrations were in the world oceans. Now, it turns out we have Secchi depth readings that go back over 100 years. So the Secchi depth was developed by a Jesuit what at that time was an astronomer. We just lower a plate into the water and determine when you can no longer see it, right? That's one optical depth by definition. So as the optical depth is only changing in the ocean because of particles in the upper ocean for the most part, you can use the optical depth history to determine whether the chlorophyll concentration of the world oceans have changed. And in the Pacific Ocean, the French and the Japanese had made many thousands of Secchi depth measurements from around 1900 to when I stopped measuring or looking at the measurements around 1990 or so. And the answer is that the ring around the ocean there, the gyre got a little bit greener over time, and the center of the gyre, the North Pacific, got a little bit bluer. So you had a little bit more chlorophyll concentration around the ring of the gyre. So why should that be? So I told Suki, I think the obvious answer is the gyre is rotating a little bit faster because the equator to pole heat gradient is greater. And you have more wind. And Suki said, nah, you can't say that. There's no evidence for that. Well, there is actually a little bit of evidence for it, but I didn't put it into the Nature paper. And you know, so I think that's an example. If you go back and look at those data, they're online in the Nature paper. And you can take a look at, and the chlorophyll concentrations from space are very similar to a Secchi depth measurement. Actually, quantitatively, you can prove that. So we have observations that now span roughly 120 years for the major gyres of the world. We do not have Secchi depth measurements that are consistent for the North Atlantic. The British and the Americans specifically made Secchi depths classified because of you could detect submarines with them. So they are possibly unclassified now because we have much better mechanisms, but I haven't gone into the data bank to look. But that could be a good subject for a student if you wanted to do that.
Okay, thanks.
Hi, Paul. I'm an ocean biogeochemical modeler, and several times I've been to the ocean productivity site, the Oregon State University, and there are different NPP products. And I was wondering if you had to give advice to a modeler as to which algorithm, like there is CAFE, VGPM, CVPM, Epley, CAR.
No, don't use the Epley model.
No.
Okay.
Let's say CAFE, VGPM, and CVPM.
Yeah.
I guess those are the three that are mostly used. But if
Right.
you have one, which one would you pick up and
Well,
why?
the one that is used now, I mean, how good it is. Obviously, we use the VGPM. That's the original model. Mike has tweaked it a bit. You know, for consistency's purposes here, I still think it's important if you're going to compare models, compare them with actual data from the ocean. You can't compare models to models. So the differences are whether you're measuring the carbon in the ocean, which Mike tries to do using optical measurements of backscatter, or if the temperature correction, which is a little bit hairy in the VGPM, is accurate, and you know, it's an empirical observation, obviously. And that's the difference. I think if you go back and look, if you can, you'll see why the Epley model is wrong. So when Epley did temperature and photosynthesis in the sea, or growth in the sea, I think it was, that was the paper, temperature and growth in the sea. It was published in the Canadian Journal of Fisheries Science. He plotted the effect of temperature on growth rates from all kinds of laboratory cultures around the world, all. And then he fit an exponential function to it. The exponential function that he fit was to the maximum growth rates, not to the observed growth rates. Virtually every single point on that curve is below that exponential line. So that's a huge problem. So it's actually an interesting problem of how do phytoplankton respond to temperature. It's obviously not linear. So you're going to have different groups of phytoplankton that will do better at higher temperatures, specifically dinoflagellates. And you're going to have some that'll do a little worse, like diatoms. But that's not what we try to do in our general models of productivity. It's an interesting question for the Margalef world, if you will. Anything else? Going once? Going twice?
Actually, I want to ask a question about the FVFM. I'm wondering if you have ever tested the FVFM in different organisms in the lab, because
Sure.
maybe diatom, coccolithophore, and cyanobacteria have different level of the number of the FVFM.
No, it's actually not true. So it's relatively simple. The reason the FV over FM is virtually constant in every single organism is because the core reaction center of photosynthesis is the same in a cyanobacteria and a diatom and an oak tree.
You think it's
And so this is a paper we published in Plant Physiology of all places, I think in 1986. It was Kolber's first paper with me, and John Zier was in the lab, and we did this with many different species. And I subsequently published it with many different species in Biochemica Biophysica Acta. So they're not in the oceanographic literature, they're in biophysics literature. Because I wanted to see whether I could convince myself biophysically that FV over FM is virtually constant. And the number is 0.65, the maximum FV over FM for a single turnover flash. If you're using a Waltz fluorometer, you're going to get a number that's higher because they're not single turnover flashes usually. You're going to get a number that's on the order of about 0.7, 0.72.
And do you think that number is mainly related with the concentration of the chlorophyll or the composition
No,
of the
not at all. Totally independent. But if you're going to measure chlorophyll variable fluorescence, you must do it in an optically thin suspension, because if you do it, if the suspension is too thick, the chlorophyll fluorescence is reabsorbed within the cuvette, and you get absolutely the wrong numbers. There's an awful lot of garbage in the literature, really bad measurements. I mean, one of the classic bad ones was using an old Waltz fluorometer. There were a group of people that went to sea and they couldn't detect the fluorescence because the instrument is not sensitive or wasn't. And they filtered the cells and put the filter into a cuvette, and total nonsense. You cannot do that. So the optics of a filter are totally different from a suspension.
And what do you think the composition, like chlorophyll A, chlorophyll is the main pigment in the ocean? Would they change the FVFM?
No. So all chlorophyll, no chlorophyll B doesn't fluoresce in a cell unless you extract it with an acetone or some solvent. All the energy from chlorophyll B and chlorophyll C, they're all transferred to chlorophyll A. So you're only looking at chlorophyll A. The only time that doesn't work is when you have phycobilisomes, and then you can have some fluorescence of phycobilisomes. But that's not what I'm measuring with FV over FM in this particular case. Okay.
And do you think, since it's a kind of indicator for the PS2, do you think this PS2 can change from time to time or from different environment?
No, it doesn't change. It's an incredibly conserved, highly conserved machine. They're virtually, so, you know, in the core of PS2 has several proteins, but D1 and D2, D1 is 86% conserved across in sequence across all species. We have the structures of these in the protein data bank at 1.2 angstroms resolution. And the structures are identical for basically for a diatom, cyanobacteria, and for, as I said, an oak tree. So you can look for yourself.
Okay, thank you.
Okay, thank you. Okay, thank you all. Nice chatting with you.
Thanks, Paul. Just once more to thank you for the talk.
Okay, thank you. All right, bye-bye.
Will you join us in an hour and a half or so for the?
I will
open-
try. I will try.
Okay, we'll be.
You'll
we
send
are-
me a link?
Sorry?
It's the same link?
Okay, yeah, Katerina will send you a new link to connect
Okay.
in an hour and a half. Gone. Is that right? Yeah, well, I hope he got the message. Well, just send him. Okay, now, maybe I think we can all stay here. Right? We just kind of stay here for this 5 group, right? So we probably kind of just join with your group, so every group. And the idea now, we have now an hour or so. So the first half an hour, the groups that are not leading the discussion, but all of you have to kind of try to share your notes and your thoughts about the whole way and the photo, and try to kind of run the kind of what we remark, the result, and the whole thing together. And this first half an hour, the group that will leading the discussion, we have to send the question, the remark, the main point of the photo to the discussion group. We're going to do that through what email, Ramiro? We will need the email address.
By email to
One
one of the persons that
we can write the email there. Well, yeah, but it will be cool that every group kind of summarizes the issues and send it to
the meeting goes to main company.
Yeah, so we can. Can we write down the email here? Yeah, or there? Yeah.
Yes.
That will go for an hour, and then we will have coffee.